I'm John, here with Manda. Thank you for inviting us. We'd like to talk today about a genome engineering technology developed in our lab, that is David Liu's lab at the Broad, which we've termed base editing. This is a technology that directly edits DNA bases without going through a double-strand break intermediate or requiring delivery of a repair template. These are two things that Feng's video touched on. As Feng mentioned in his video, Cas9 is able to bind locations in the genome that match its RNA guide and catalyze a double-strand break. This double-strand break in cells is resolved in one of two ways: through templated repair, which can generate mutations matching a supplied template, for example, by changing big to little in Twinkle Twinkle Little Star, or non-templated repair generally results in deletion of part of the sequence, flanking the double-strand break. Importantly, even in the presence of a repair template, the cell still resolves a substantial number of breaks via the second pathway. While there are cases in which a deletion is desirable, many times we want to avoid deletions. So how do we maximize proper conversion of big to little while avoiding Twinkle Twinkle Star? This is the issue that's motivated the development of base editors. A little background. DNA, of course, has 4 bases. C pairs with G and A pairs with T. As many of you may also know, RNA uses a 5th base, U, rather than T, to pair with A's. Additionally, there are enzymes that convert C's to U's in both DNA and RNA in very specific contexts. So Alexis Kamora, then a postdoc in the lab, started a project with the goal of developing a cellular deaminase. The CD reaction is termed deamination, that acts on arbitrary general genomic DNA sequences. So the idea here is to convert C to U, which then forms a mismatch, which is recognized and repaired by the cellular machinery. This repair process first repairs the G to an A, forming the base pair found in RNA, and later repairs the U to a T, thus directly converting the CG base pair to TA. And the tool she actually developed to carry out this reaction is a Cas9, which has been mutated to remove and abolish its DNA cleavage activity, fused to a cytidine deaminase. This allows us to use the programmability of Cas9 to position the deaminase where it can act on specified Cs in the genome. More recently, a project headed by Nicole Gaudelli, a former postdoc in the lab, used a similar approach to convert A's to an inosine intermediate, which is then repaired to a G. Together, I think this class of tools gives us the ability to edit individual bases in the genome, which we think is potentially very powerful. While we're working on generating additional base editors, we're also beginning to apply the existing editors to biological problems. So Manda is going to tell you a bit more about how these tools could be used.
Right, so you might wonder why it's so interesting for us to be working on these base editing applications. And if you look at this pie chart of human genetic variation that is causal to disease, you can see that the vast majority of the pie goes to these single nucleotide changes. And with the current base editors that we've developed, we're actually able to revert over half of these. And as John mentioned, we're currently still working on trying to come up with new tools that can target the remainder of them. The gene editing technologies that we have right now are currently mainly being used for research purposes to edit the DNA of human cells and animal models to be able to better reflect human genetic disease so that we can study what is actually going wrong in these situations and to come up with new ways of being able to treat them. In the near future, we also hope to be able to use these tools to target the disease-causing genes within human patients themselves. So in many cases, we don't even know really enough about the diseases to come up with proper conventional therapies. But because we do know the underlying genetics that are causal to the disease, we can go in and actually permanently fix them to treat the disorder at the core of the problem. However, as we learn even more about human genetics and disease, we're identifying genetic traits that could even confer beneficial traits onto humans. For instance, we have found individuals who are immune to it's not supposed to go that way, to infectious disease, such as HIV. And we've also found certain individuals who are essentially immune to cardiovascular disease. So we could, for instance, treat at-risk individuals or even the entire population as a whole as a pre-treatment to generally improve the overall health of our population, which has significant impact for pretty obvious reasons. However, treating patients directly with tools like CRISPR and base editing right now is still quite complex, and it will probably take a few more years before we're quite there. But what is already happening right now in early clinical trials is to treat human patient tissues outside of the body. So for instance, in the case of cancer, what we're working on is trying to take patients' own immune cells outside of the body and treat them in certain ways such that they would become exquisitely sensitive to cancers. And then we can retransplant these cells back into patients and have them go find both tumors and satellite cancer cells that are roaming through the body to help clear those up. And in the future, we also hope to be able to treat other human tissues outside of the body in more controlled and safe conditions and transplant these back in. However, there's even still more that we can do with these tools by editing the DNA of both animals and plants. And this has pretty big implications in many ways. Gene editing of animals has continued to help how we can do animal research. For instance, for larger animals, they've been able to make them smaller. This is the full size of a full-grown micro-pig that is otherwise entirely healthy. But we've also been editing the genes of livestock and crops for thousands of years now. And while most of you would probably recognize this as a watermelon from a 17th century painting, many crops prior to selective breeding are nearly unrecognizable to us today, and this has been incredibly beneficial for feeding the world, among many other things. And finally, pest control is another important way where gene editing can have a significant impact on human health and the environment. Gene editing can be implemented to limit the populations of disease carrier species or of invasive species that threaten native ecosystems, while limiting the impact on the surrounding environment. So all of these processes are things that we're already currently doing. However, gene editing can aid in all of these slow and difficult processes by creating genetic variations that we're looking for within a single generation. So the application of these tools can be incredibly powerful and has the potential to greatly improve our lives in very many aspects. Thank you.